Hi, everyone. My name is Lynn Hernandez. I'm with the Desmoid Tumor Research Foundation, or DTRF. It's great to be here with you tonight for our webinar on clinical trials and a new phase 1 trial, which includes desmoid tumor patients. Before we begin, I have an important message. Some of you have submitted questions, and they have been shared with our presenter tonight. He will do the best he can to answer them. And if he's unable to speak to specific cases, you'll be patient with that. Thank you. Information is absolutely essential when it comes to managing and living with desmoid tumors. Each case is different. This conversation is not intended as medical advice for any individual problem or as a diagnosis, treatment plan, or recommendation for a particular course of action. It should not be used as a substitute for professional medical advice or services. This webinar is for educational purposes only, and the DTRF does not endorse any treatments or studies. If you have any other questions unrelated to today's webinar, please reach out directly to the foundation at info@dtrf.org, and we will do our best to direct you. We'll add our contact information in the chat as well. So this is how we're setting up today. We invite the community to post questions in the Q&A. If you have questions for our presenter, post them in the Q&A. You'll also notice the chat function, which will not be monitored for questions during this session. So attendees that want to communicate with the community, you post in the chat. If you have questions for our presenter, please post in the Q&A. If you know someone who is meant to join but isn't able to, please tell them that this recording will live on our website in our video library, and we'll notify the community when it's public. Please be sure to subscribe to our newsletter at dtrf.org for notifications. Okay, so our guest today is Dr. Gregory Cote. He's a medical oncologist with Massachusetts General Hospital Cancer Center, and his focus is phase 1 trials and sarcoma. His primary research interest is in developing novel agents for connective tissue malignancies through translational studies and early phase clinical trials. Dr. Cote, we're so grateful to have you with us. I know that you're working hard in the clinic, and we thank you so much for taking the time to teach us about the clinical trial process and also this new phase 1 trial. Welcome, and I will turn it over to you.
Great. Thanks so much for the introduction. Let me just pull up my slides real quick. All right. There we go. All right, great. Well, thank you again for the introduction. So as you heard, I'm a medical oncologist. I spend my time taking care of patients and then also running clinical trials. And in that clinical trial space, I focus on what we call like early phase trials. So when a drug first gets into patients, and then I do a little bit of later line drug development as well. So we'll talk about that as we go through. So Lynn and the team had asked me to kind of give you guys an overview of clinical trials and then to talk about this new trial we have called FOG-001 and why it might be of interest to patients with desmoid tumor. Just some housekeeping, just for disclosures, just so you know. So I'm an investigator on the study, but I don't receive any salary or support. I don't have any stake in the game, whether it's successful or not, other than just being happy that we're developing new drugs for difficult to treat tumors. And my institution also receives some funding for the conduct of the study, just to carry it out. Okay, so jumping into it. So for our outline today for this talk, we'll cover some of the challenges in developing drugs for rare tumors like desmoid. I'll give you a basic primer on how clinical trials work, and we'll focus in on that phase 1 part. And then I'm going to take you back to high school biology. We're going to talk a little bit about cell signaling and how it relates to desmoid tumors, because that's a good setup for how this drug works. And then we'll talk about the trial. And then, assuming I don't talk too much and we have time, I'll give you an example roadmap of how I think about patients with desmoid tumors. All right, so let's jump right into it. So I'm sure this group knows well that desmoid tumors are rare. So it's about 5 cases per million people per year. So just think about that in contrast to something like breast cancer, where there's like 100,000 cases per year. You might imagine it'd be hard to run studies when you have a rare patient population versus something that's more common. The other challenge here is that we see what we call a heterogeneous clinical behavior. This is where some desmoid tumors may grow very slowly over time, maybe a few millimeters every few months, and not really be a huge clinical problem. Others can grow quickly and then spontaneously regress. We don't really understand why that happens. And then others can kind of grow and grow and grow. And so you can imagine if you're trying to develop a clinical trial for this, if you accrue a bunch of patients that are kind of more slow growing or spontaneous regressing, you might think that your drug is better than it actually is. And so it kind of forces us, even though this is a rare population, to think about designs that might involve placebos, even though we try to avoid that where we can for rare diseases. This is probably less true today than it has been in the past, but for any rare disease, whether it's a rare tumor or rare cancer, it's a lower priority for drug companies, and this is really a financial consideration. There's a smaller market to recoup the investment, and that's been difficult for us for a lot of rare cancers. But fortunately for the desmoid community, we've had a couple of drugs get through to FDA approvals, and perhaps some more on the way, which has been great. The other thing that we can sometimes capitalize on is that for pathways such as this beta-catenin pathway that I'm going to talk to you about, it's actually important for other cancers. So more common cancers like colon cancers. So we can do what I call like a Me Too approach, where, yeah, we do this drug development in this common cancer, but let's also think about rare diseases where that pathway is important. So we'll get into that a little later as well. And then this is I think an important consideration for desmoid is that all of our current therapies, either approved or not, really rely on an indirect approach to treat the tumor. And what I mean here is that chemo is designed to kind of disrupt DNA and stop the cells from growing. Some of these tyrosine kinase inhibitors block some important proteins that may be important for those cells. And these new gamma secretase inhibitors also block a pathway that may be important for those cells, but it's not really getting at the exact biology of the disease. And so that's been a challenge. And so there's been some interest in whether we can target this beta-catenin, this pathway, and as I said. So taking that all together, there's a high interest both in the patient community and on the research side in developing drugs that are more effective. And I honestly think that someday we will be able to treat this and maybe even cure this disease. But if we can't do that, we want to make sure that these therapies are tolerable. So if somebody's going to be taking them for a long period of time, we want it to be as low side effect burden as possible. So when we think about developing new drugs, these are high priorities for us. All right, so let's pivot here and talk about clinical trials. So what is a clinical trial? So in the oncology space, this is where we're trying to match a new therapy with a certain patient population. Hopefully you can see my mouse here. So this is mostly done for cancers, but we also have different tumors and that are not cancers, meaning they don't metastasize to other areas of the body, but can cause a lot of problems. And we need to develop treatments for them as well. So we're trying to match these two together. And this is a really complicated process. This is a high-level view of what a clinical trial is. It's sort of the lifespan of a drug, if you will. So things start here in this preclinical research phase. So this is where we have maybe a new drug we're interested in, and we're testing it in like a petri dish with different models of cancer. And if things look good and it's doing what we want it to do, we might test it in some animal models and show that we can stop the growth of that tumor or that cancer. And if things look really good, then there's a decision that's made, and we want to bring this drug into the clinic. And that's what we call phase 1 clinical trials. So this could be a company, could be a big pharma company, could be a biotech, could be an academic university, could be a National Cancer Institute. Any of these things can run clinical trials. So when we do these first-in-human studies, phase 1, our goal here is really trying to figure out, is this drug safe and what's the best dose? This may look great in a mouse, and it may crush the tumors and they all tolerate it perfectly, but mice aren't human, and we need to know what's the right dose, what are the side effects, and importantly, can it start to treat some tumors? So as we move from phase 1, we go to phase 2, and phase 2 is typically where we have a patient population that's uniform, could be all the same diagnosis or maybe they all have the same mutation, and they're typically treated all at the same dose. And we're really trying to build a story here that we're actually seeing some effect of treating these cancers or tumors the way we want to, perhaps compare it to some historical control. As this drug matures, we move on to these later phase studies, and this is where we're comparing the drug versus some other prior treatment or perhaps a placebo, and we're trying to prove that it's actually better than what we have. And this is a good example of this would be like the nirogacestat phase 3 study. So that study was a randomized study. So patients were blinded. They either 1-to-1 received either the drug or placebo, and we were able to show that the nirogacestat was superior for treating desmoid patients, and that ultimately led to its FDA approval. Now, you're probably looking at this and saying, okay, this is a lot of time. You're absolutely right. This can take up to 10 years, could be billions of dollars, and hundreds of patients. And so there's this huge pressure to try to figure out ways to be more efficient here in this early phase part of the studies to really make what we call a no-go decision. And the idea here is like, it used to be the old priority here was just what's the right dose and is it safe? You might be able to figure this out with a handful of patients, but now we really want to see, like, is this actually treating the tumor that we think it is, to make a decision to whether we're going to go further. So what we're seeing now is that there's a lot of studies where phase 1 and phase 2 are being combined. So we have the first part where we figure out what's the right dose and what's the side effects. And the second part, we're kind of expanding within that same study to really show that it's actually working. All right, so let's take a deeper dive into phase 1. So again, as I told you, we don't know the dose when we start this. And so there's a lot of different models here on how to figure out that dose. And the most classic design is something called a 3+3 design. And the way this works is we pick some starting dose level 1, and that's modeled off of prior animal experience. And we actually only treat 3 patients at a time. We follow those patients at this low dose for some predefined specified time, usually it's 3 or 4 weeks, and we see how they do. And the way this study works is if there's certain rules, but if in this example, if less than 1 out of 3 experiences a severe side effect from the drug that we think is related to the drug, then we're allowed to increase the dose. So in this cartoon example, we follow 3 patients, they do okay. We increase the dose and we treat 3 more patients. See how they do? They do okay, we increase the dose. And eventually we reach a point where somebody has a bad side effect. And we say, okay, we got to pause there, and we kind of look at what happened. And as long as we feel like it's safe to do so, we'll add 3 more patients. So that's the 3+3. So these next 3 patients do okay. So we're still below 1 and 3 for our side effect events rate that we threshold that we started ahead of time. And then from there, we kind of keep going and going and going. And eventually you reach a point where you hit this threshold of 1 out of 3 patients in the 3+3 design. So at this point, there's a pause, and all the investigators and the scientists who are working on the study kind of look at all the side effects, look at the efficacy data. So is this hitting the target that we want? All during this study, we are measuring levels of the drug in people's blood, and that helps us kind of characterize our dosing to make sure that we're dosing it correctly. And we'll take all this together and we'll determine what we call the recommended phase 2 dose. So this is the dose where if we go to the second part here, we're going to take it from phase 1 to phase 2. So we go through this process, and then most studies will have something built into this called a dose expansion, where basically it's like a small phase 2 study where we're just treating more people at the dose we pick. So say we pick this dose level 4 to do our dose expansion, we're going to treat another dozen or another 20 patients to make sure that we're seeing the safety that we expect from that dose level. Now, all through this, you're probably wondering, what about activity? Like, what about anti-tumor, anti-cancer activity? And as I said, this is a really important part of these early phase studies. And we're, as investigators, we're really looking to see that we're going to see that anti-tumor effect that we're looking for to make a decision to go to phase 2. If we treat all these patients and we're not seeing any activity, we're going to pump the brakes a bit here and say, maybe this is the wrong plan. Now, one thing I didn't mention earlier is that of all these patients, typically these are all different kinds of cancer or tumor patients. It's usually a mix. For a drug that's maybe a chemotherapy drug where we don't have a particular population we're looking at, it may be all different kinds of cancers. If it's a particular drug that's targeted to a very specific mutation, maybe we just include different kinds of cancers that have that mutation. If it's a particular drug that we think is just going to work in lung cancer, we might run this phase 1 trial just in lung cancer. So it's a really important thing to think about. And in some ways, it also lets us kind of explore some of these novel drugs that maybe the company's interested in developing for a different, more common cancer, but we can kind of sneak in some of our rare tumors into these cohorts. All right, so we go through this process and we come up with our phase 2 dose. Now, you're probably asking yourself, what's the balance here of the potential toxicity versus trying somebody at a higher dose versus a low risk of side effects at a lower dose of drug that maybe isn't active? And we struggle with this as oncologists because we may be excited about a drug and a particular mutation, but we worry a bit at this lowest dose level. Are we going to see the activity that we want? At the same time, I have somebody I'm treating at the highest dose level. We've already seen some side effects and some borderline tolerability. Like, I'll be nervous about the side effects that patient may experience. And so this is something that we have to kind of weigh as we kind of make these decisions on what we're going to do for an individual patient decision. Now, I will say that if you do this long enough, and I have been, for the right drug and the right target and the right patient, we actually do see activity even at these dose levels. And I've been lucky enough for a few different trials to see amazing responses in these first dose levels. And like I said, if the science is good and the target's good, you can see that effect. So we'll put people on, but it's a conversation to be had with your oncologist if you're considering a trial like this. All right, so moving on. So we talked about phase 1. So phase 2 again is we figured out the dose from phase 1, and now we're jumping into phase 2. So we have the dose determined from phase 1, and as I said earlier, phase 1 is a mix of different kinds of patients, cancer patients or tumor patients. The phase 2 is typically everybody's aligned. You try to get patients as similar as possible together to really kind of build the evidence that you're seeing the level of activity that you want to see to make this jump to phase 3. Because what we don't want to do is have a kind of a mixed signal, go into phase 3, spend a ton of money, waste a bunch of patients' time on a drug that may not work. So we really want to go into phase 3 being pretty confident that we have the right drug for the right indication. All right, that's confusing. Hopefully, I didn't lose everyone. All right, let's talk about what happens next. So you've signed on to a clinical trial, or you're interested in a clinical trial, and you want to talk to your doctor more about it. So what typically will happen is you'll meet with your oncologist, and they may say to you, I have these things that are standard of care, and I have this clinical trial. This clinical trial is about this. Is it something you're interested in? Talk to you about it at a high level. And if you are interested, they'll go through something called informed consent. So what's informed consent? So a consent form for these clinical trials is a 20 to 30 page document. It's pretty thick. It's written in lay language, but the goal here is to really explain to you what the trial is about, who's funding it, why they're doing it, what the design is. Is this phase 1? Is it phase 2? Is it phase 3? What's expected of you? Like, when do you have to be there? What's expected of us as the docs? And then what are the side effects of the drug, and what's the schedule that you'd have to be there? So it's a lot to go through, but it's really important. And now you may go through this with your oncologist and say, thanks, but no thanks, doc. This doesn't really seem like it's right for me, and that's okay. Like, your oncologist will still be your oncologist. They're not, not going to kick you to the door. But if you are interested, then you'll go through and you'll do this signing process. But again, it's really important to recognize that this is your choice whether you participate or not. If you do sign on the dotted line, the next part is called screening. And screening is where the trial has this required tests to make sure that you're what we call eligible for the study. So it's basically like a checklist to go through to make sure that there's no issues that ahead of time might be risky for you to take part in the study. So, like, good examples will be if this study's for lung cancer and you have desmoid tumor, you're not eligible because you have the wrong kind of cancer. If you wanted to participate in the study, but you have maybe kidney or liver injury and the drug is metabolized through the kidneys or liver, then it might not be a good fit. So these are exclusions that you might not be able to participate in. On the flip side, though, if you have the right tumor and your organ function is good and the labs are good, then you will cruise right through. So once screening's done, then you start the treatment part of the study. Now, every study is different, and whether it's a pill or IV will depend on that particular study, what the schedule is. Do you have to go there, every week, every 3 weeks? That will all be dependent on the particular study. So these are really important questions to ask when you go through the consenting process, because there could be an issue where you're interested in the study, but logistically you just can't make it work because it's you got to work and it requires you to be there so many days per month. So we'll work with you on that and just make sure you ask those types of questions. Okay, so key points here. So informed consent, I can't emphasize how important this is. You're not a guinea pig, you're an active participant in the study, and you need to be involved in this process for deciding if you do it or not. Early phase are these first in human studies. They tend to be very busy, lots of extra visits where we're checking for safety. We might be measuring the level of the drug in you. We're doing EKGs. Sometimes there could be required biopsies because we're interested in sort of the biology of how this drug hits a target or not. And that's really important for the drug development. The flip side is you get access to cutting edge technology that it wouldn't be otherwise available. And there is no placebo in early phase studies. For later phase, they tend to be less busy because we have more prior experience with the drug. We already know the safety profile. So we may be confident that you only need labs checking once per month, that kind of thing. But the flip side there is it could be you could be randomized to a placebo arm or something like that. All right, so that's a lot of information. We'll happy to take a lot of questions around that because I realize it's confusing and it's an important part, but part of the talk. But I want to make sure that everybody's got a good sense of what this involves. All right, we're going to pivot now, and we're going to take you back to high school. We're going to talk about cell signaling. And the goal here is just to give you an update or a background on how desmoid tumors are driven, like what's the biology behind them and how we can think about drugging those. All right, these are my cartoons. This works much better on a chalkboard, but I drew this a few nights ago, so hopefully you'll be able to follow. So this is a cell. This is a normal cell. And outside of the cell's world is out here, inside is in here. And inside the cell, there's this thing called a nucleus. And inside the nucleus is your DNA. This is your genetic programming. It's basically the coding that makes you, and not somebody else. Now, every cell in your body has the exact same DNA, which is crazy to think about. But a skin cell looks different than a muscle cell, looks different than a fat cell. So how can that be? So it turns out that there's different programs and different gene programs that are turned on and off in the life of that cell. So if your cell is destined to be a fat cell, only those genes that are important for the fat cell are turned on within the nucleus, and everything else is shut down. Interestingly, cells can interact with their outside world. And so if they need to get a signal to grow, so say it's a skin cell, you've been injured, you need to grow some skin back, they actually can respond to external stimuli. And so these are called ligands, and I made a little square here, and they bind to something called a receptor on that cell to start this signaling process. So basically, that ligand will hit the receptor, and then it starts this signaling cascade within cells. And there's many steps. I just simplified this into a couple, but there's many steps that are involved in this, and that can tell that cell to grow. It turns on that machinery. And then there's important inhibitory pathways, and there's a lot of different mechanisms for this. Some of them will block the signals. Some of them will target some of these things to be degraded, send it to a wastebasket recycling program. And it's incredibly important that you carefully regulate all of these. And that's how we avoid cells growing and becoming tumors, right? So the idea is that even if something's misfiring, these other pathways can try to regulate this process. So when we think about cancer and tumors, something's gone wrong in this process. There's different mechanisms of how cancers can form, but this is sort of a simplistic idea of some tumors and cancers that have pathway addiction, we call it. So something on this pathway has a mutation. So even in the absence of ligand, this pathway is stuck in the on position, and it overwhelms all the inhibitory programs and tells this cell to grow. Similarly, you can have a mutation in the pathway that leads to recycling and degrades this. So now, even though there's no signal to keep things going, this is just stuck in the on system because there's no inhibitory signal. And really, like any of these parts of this pathway could be broken along the way. You could have too much ligand. You could have a mutation in the receptor. You could have some mutation within the nucleus that can lead to these pathways being stuck in the on position where they shouldn't be. So when you hear this term targeted therapy, this is where we're developing drugs to target something that's gone wrong in this pathway. All right, so let's bring this back to desmoid. So the Wnt beta-catenin pathway you may have heard of. This is the pathway that is abnormal in most desmoid tumors. And in a normal cell, again, normal here, when this is off, the ligand is called Wnt, and it binds to a receptor that leads to activation of this pathway. So when this is off, these two proteins, APC, which can be mutated in desmoid, and beta-catenin, which can be mutated in desmoid, are tightly bound to this protein that basically targets beta-catenin for recycling. So when the ligand's not present, beta-catenin can't go to the nucleus because it's been rerouted to get recycled. When the ligand binds, there's some changes that happen in these two proteins. The recycling program falls off, and then beta-catenin goes to the nucleus where it binds another protein and turns on genes that are important for that pathway. All right, so off, it's degraded, on, it gets to the nucleus. Okay, so let's come back to desmoid. So what happens here? So there's two mechanisms here. So we can have mutations in APC that lead to disassociation of this recycling program, and then a beta-catenin accumulates and goes to the nucleus. Or we can have a mutation in beta-catenin where, again, this can't bind to it and it leads to accumulation. And so basically, you have all this beta-catenin swimming around, and I can't stop it because it's stuck. Now, there are, Lynn and I were just talking about this, there are rare cases of desmoid where we don't see either beta-catenin or APC mutations. And you have to wonder, is there another mechanism that we don't understand, or is there maybe a mutation that's just not captured well in our genetic testing, or is there some other pathway? We don't know. So there are other ways to activate and inactivate proteins that we don't fully understand, and it could be a different mechanism like that. All right, so bringing this back to the FOG drug. So what's this? So this is a drug. So this is another cartoon of beta-catenin binding DNA. So it binds this other protein called a transcription factor, and together they turn on DNA. So this little up arrow is a cartoon for biologists to say that the gene is turned on. And what FOG-001 does is it actually binds between these two proteins and stops this interaction. So even though we have beta-catenin swimming around everywhere in the nucleus, it can't do anything because this drug disrupts that. That's the concept. So bringing it back home. So this trial is a first-in-human study of FOG-001 in locally advanced or metastatic solid tumors. So what does this mean? So they're basically interested in different kinds of tumors, and it's a phase 1 study. We'll jump into some details on that in the next slide. Clinicaltrials.gov, I wanted to touch on this because this is very important for you. So this is a website that's run by the government, and any trial that is open in the United States has to register there. And on it will say what the indication is for, what the eligibility criteria are, where it's open, and if it's open or if it's closed, that kind of thing. So you can actually go onto this website and search desmoid tumor recruiting, and it will give you all the studies that are open for this tumor. If you have friends with other kinds of, or family with other kinds of cancers, you can do that as well. You could look in colon cancer, like open and recruiting. It'll tell you the sponsor. So in this case, this company Parabolis is developing this drug and providing the funding for the study. And it will tell you when it was last updated. So let's jump to the next study. Okay, so this is a phase 1/2 study. So getting back to what I was saying earlier, so the phase 1 part is where we're trying to find the right dose, what are the side effects, and the phase 2 is once we've found that dose, we're going to treat more patients at that dose. The drug is given weekly by IV, and in this study, there's different cohorts for treatment alone, and then at some point, there's also interest in combining this with other drugs for different kinds of cancer, not for desmoid tumor. Yeah, and so just highlighting here, we're at this part of the clinical trial schema that I showed you. If things are successful, we hope that we'll go to later phase, but we're still in this early part right now. Okay, so we covered some of this already, but I just want to take a minute just to go through it just to make sure everyone's on the same page. So phase 1/2 we talked about. Multi-center just means that it's open at multiple different sites. I have a map of that in a second. Open label non-randomized means that everybody who's getting the drug knows what they're getting. It's not like randomized to placebo, nothing's blinded. Sometimes for those phase 3 studies, it's blinded, so I don't know what the patient's getting and the patient doesn't know. And then we just find out at the end what they were actually getting. So everybody's getting the drug, everybody knows what they're getting. Dose finding gets at this phase 1 part that we talked about earlier, and then it's given IV alone or in combination for different cohorts. So for right now, we're in this dose finding, dose escalation part where we're trying to find the right dose. But once we've established that, then we're going to do dose expansion where we treat more patients at that dose, similar to what I said earlier. And then this part is really just to highlight a few things of the study. So if you focus on the right side first, so there's different cohorts for different types of tumors. So there's a group of patients that have desmoid tumor that is the focus of what I'm talking about now. But there's also patients with other cancers, including colorectal cancer, where they're also interested in developing this drug. It turns out this beta-catenin pathway is very important for colon cancer. I might have mentioned that earlier. So it's being looked at in that area as well. Later on, the company's interested in combining this with other different chemotherapy drugs, but this would again be for non-desmoid patients. We talked a little bit earlier about inclusion and exclusion criteria. This is part of the screening process. So for this particular study, there's a long laundry list, but these are just the highlights. You have to be over 18. We often do that for early phase studies just because we want to, we don't want to treat any vulnerable populations such as children with the unknowns that are built into that study. Performance status is really how fit you are. This is not typically an issue for desmoid patients, but for cancer patients, if you're less fit, there's a higher risk of maybe a bad side effect being really bad. And so this is really just a calculation of how fit you are, where normal is 0 and 1 is maybe a little bit of fatigue. Adequate organ and bone marrow function is just making sure that things will be safe. And then there's some exclusions. Probably the most relevant for us in this conversation is that this beta-catenin pathway is actually very important for bone metabolism. So for patients who have osteoporosis, they're excluded, unfortunately, just because we don't want to make that worse. All right, so I think we talked about the rest of that. So this is the map of where the study's open. So I know there's some international people who are interested in this. Right now, it's just open in the United States. There's some decent coverage across the US. I don't know for sure, but usually as these drugs kind of mature, there will be international sites that will open. So hopefully that will be the case for the drug as well. All right, so this is a question I wanted to kind of head off at the pass. How do you know an early phase study is right for me? So this is a really difficult calculation for a desmoid tumor, which is like I said, not a cancer. It's an intermediate grade tumor where it's not going to metastasize and travel to other areas of the body, but it can cause a lot of suffering. So this calculation is a little easier for a patient with an advanced cancer that's traveled to other parts of the body and they've been on other treatments that haven't worked. It's a little harder for the desmoid population where these tumors aren't typically fatal, right? They're usually in locations that we can manage. So weighing through the pros and cons, it gives you access to cutting edge technology. As I said earlier, this is a targeted therapy. Like, I put in here laser focus on the pathophysiology of this disease. Like it really hits there. The other drugs that we have, gamma secretase inhibitors, tyrosine kinase inhibitors, chemotherapy, these are really indirect methods of attacking that pathway. This could be a pro or con, depending on your personality, but the research teams for early phase studies are really involved in your care. You're going to get a lot of attention. There's the investigator, co-investigators, research nurses, research assistants, and you'll find that you'll have a lot of people checking in on you. And the reason we do this is because there's so many unknowns about these drugs that we want to make sure that you're safe. So there's frequent check-ins, a lot of phone calls. And if you're somebody who values that kind of attention, then you're going to love this. If not, then it might be a little too much. So you have to think about that. And then getting at what I was saying at the beginning, other treatments may not have worked for you, or maybe you've had bad side effects and you're looking for something new, like this could be a good opportunity for you. The flip side, though, as I said, very busy schedule, a lot of extra visits. There's a lot of unknowns about the early and late side effects. That's true with any new drug. uric acid, sorafenib, liposomal doxorubicin, these all went through the same process. And when those drugs started, we didn't know a lot of the side effects. So you just have to keep that in mind. There could be biopsies, as I mentioned earlier. There's definitely going to be extra blood draws and EKGs. These are part of, required to do this as we develop drugs to really characterize how these drugs develop. So you got to think about this and kind of weigh the pros and cons. And if it's right for you, then you can talk to your doc about participating in something like this. If you think it's not quite right and you want to wait and see how the drug matures, that's totally appropriate and very reasonable. All right, so let's just spend the last 90 seconds here and talk about how I tend to think of a new patient or a recurrent patient with desmoid. I think, if they haven't seen me or somebody at my institution before, I generally want to confirm the pathology. I'd say, 90+% of the time, the outside pathology is confirmed that it is a desmoid tumor. Every now and then we get a low-grade sarcoma that was confused to be a desmoid tumor. So I like to make sure the pathology is confirmed with pathologists who are experts in that area. If you're followed in the community, that's totally fine. There's a lot of amazing community oncologists who can certainly manage desmoid tumor. I like to have people see an expert sarcoma medical oncologist at least once in their pathway, just somebody who can make sure that they're on the right track and they're not doing something that maybe is outside of what we would recommend with current guidelines. And then this is sort of a tough question for desmoid, but something worth thinking about. So most desmoid tumor are driven by beta-catenin mutations that are mutated within that cell. There's a subset that can have mutations in that APC gene that I mentioned earlier, and that can be inherited. And it's really important because it can be passed from generation to generation, but it also puts patients at risk for other kinds of cancers, including colon cancer. So there's a certain calculation, and it's worth a discussion with your oncologist of whether you should have genetic testing or not. Usually this is in somebody who's younger, maybe somebody who has a strong family history of colon cancer. These are situations where I wouldn't want to miss that. All right, the next question is treatment indicated? So I would say like 85-ish% of my patients, I can actually just watch them and their tumors, maybe it's there, maybe it's growing slowly, but it's not causing any major symptoms. And, that's an ideal situation. And we'll just follow them over time, set up some kind of schedule for follow-up, maybe imaging every, 6 months, every year. Sometimes patients are, fine. And they say, you know what, I'll just call you if it starts bothering me. That's totally appropriate. If patients do need treatment, though, we have to think of one of these different drugs. And this is I'd say like a shared decision-making, because we have lots of options and they all have kind of different pros and cons. And, depending on the individual features of your situation, we may think about one drug or one opportunity versus another. These could include these gamma secretase inhibitors like uric acid, tyrosine kinase inhibitors like sorafenib, chemotherapy in some cases, clinical trials if we have options. And then there's other things like ablation. And then rarely, but occasionally we still do this, we'll think about surgery or even radiation for the right situation. So we'll talk about all these. And then, you're not going to be on this treatment indefinitely. In my mind, we start one of these treatments until we can knock the tumor down to make it stop growing, stop causing symptoms, and then we'll take a break. So maybe you'd be on the treatment for 6 months, maybe 12 months, but then we take some pause. If it starts growing again and causing symptoms again, then we kind of got to go through this math. Do we have to, should we try something new again? And we kind of go through this process. But ultimately, the hope is that we can kind of quiet these down enough where maybe they're there, but they're not causing a problem. We can just watch you over time. So, your roadmap will look different than this, but this is just an example of what I was thinking of. Maybe you start with some observation, you go on the uric acid or some other gamma secretase inhibitor for a period of time, that stops working, or you have side effects. Maybe you try that again, or maybe you go to sorafenib. Maybe you do a clinical trial, maybe we do chemo, maybe we do an ablation, and you kind of just march down this line over time. And hopefully you reach that point, as I said, where the tumor is quiet and not causing symptoms and not a problem for you medically. So I think that's my last slide. Yeah, so why don't I stop there? And then I know we've got a bunch of questions and we can try to get through them with the time that's left. Oh, Lynn, you're muted. Lynn, you're muted.
Sorry about that. Thank you. I appreciate that. Thanks so much for all this great information, Dr. Cote. We definitely do have some questions. So just in general, with regard to clinical trials, can international patients coming from outside of the US, apply to clinical trials within the US?
Yeah, so this is a complicated question. So the funding for clinical trials in the US is a little different than some parts of the world. So anything on a trial that's considered standard of care, so routine blood draws, routine scans, visit with me, things like that, that's covered by insurance. Anything that's considered pure research, so this could be the drug, extra research lab tests, biopsies, things like that, that's covered by the trial. So if you're in the US and you have insurance, be it private insurance or Medicare, Medicaid, they'll cover trials typically. We have to, we have a team that actually will, most places have this, that will clear your insurance before you participate. But if you're international, then it gets a little tricky. So your options are really either you have international insurance that will cover something like this, or you have to self-pay for the parts that are not considered research, and that could be very pricey. So I'd say it's feasible, but often at a very high cost, so not feasible for everyone. Now, the National Cancer Institute, they as far as I'm aware, do not have any desmoid-specific trials open right now, but if they have a trial that's of interest and you're eligible for that, they actually do cover the cost for international patients, last I heard.
Thank you. And then there are some questions about this specific trial, the FOG trial. And you may have touched on this. I'm sorry if I'm repeating. So can patients that have FAP participate in this trial?
Yeah, great question. So FAP is that inherited mutation in that APC gene that I showed you earlier. And so patients who have APC mutations, germline or acquired, or beta-catenin mutations can all participate in the study. So it doesn't matter whether you have germline or not, you're just required to have one of those two mutations. For the rare desmoid patient that does not have one of those mutations, I think as the language sits now, you would not be eligible. We would need to think about that a little bit, probably talk to the company. It may not be that your desmoid is driven by those, so maybe this isn't the best trial.
Okay. And what is the cost of this medication, of participating in this trial? I'm sorry.
Yeah, yeah, getting back to what I was saying earlier. So anything that's standard of care is covered by your insurance. So if you have copays or maximum out-of-pocket deductibles that you have to reach, you're still responsible for that. Most patients undergoing treatment will hit that deductible pretty quickly. But anything that's otherwise research is covered by the trial. Now, I don't offhand remember the details for this study, but some studies will have a travel stipend for, particularly for these early phase studies that are very busy. So they may, cover meals or some gas. Sometimes they cover a hotel room for the long days. So it just depends on the individual study, and when you talk to your research team, they can kind of go through the logistics of that.
Okay. How does this specific drug differ from other treatments like Nexavar and nirogacestat?
Yeah, so this drug, as I showed you in that cartoon when we were in biology class, binds directly to beta-catenin. It interferes with how it binds to DNA. So that's a completely different mechanism than nirogacestat and sorafenib. So nirogacestat inhibits a pathway, but it's a different pathway called gamma secretase. And we think that indirectly ties into this beta-catenin pathway. We actually don't fully understand the exact biology, but it blocks it enough that it stops the growth of these tumors. Nexavar or sorafenib, these are what we call a dirty tyrosine kinase inhibitor. So tyrosine kinases are important amino acids that are in many different proteins in cells, and that enzymatic process is how cells function. So that signaling cascade that I was telling you about, those are all enzymes that kind of send that signal to the next. And the way those drugs work is it blocks these kind of indiscriminately across cells, and it just blocks enough of them that it's actually lethal to the desmoid cell and can stop them from growing. So short story is very different. One's like laser focused on the mechanism, and the other has more indirect effects.
Okay. Do you know the answer to this? When was the FOG medication first administered in human?
Oh, I don't know. I don't know the answer to this one. I think it's been a couple of years. You know, there's been some patients dosed for quite a bit now as they've been trying to find the right dose across many cancers, but I think it's a couple of years in.
Okay.
These
Okay.
studies, you know, they take a long time. So when I was showing you that cartoon of the 3+3, you know, every group of patients is another month to 6 weeks, and then you go to another group, and then there can be some pauses. So it's a really long process to optimize the dose and try to find it. I mean, you know, it takes some time.
Got it. Got it. Can you share a little bit about the major side effects or the side effects from this specific medication?
Yeah, I can't right now because there's not been a lot publicly shared for this drug in the study that will be presented at a research meeting at some point in the future, and then we can talk about it freely. For people who are signing on to the study, when you meet with your treating doc, your treating investigator, they'll go through all the known side effects. It's really important though to know that this is just a snapshot in time. Like you know, these are the side effects we know about now, but because this is a relatively new drug, there'll be other side effects that will evolve over time as we treat more patients. So right now, unfortunately, I can't share a lot, but you know, as more is known and more is public about this drug, we can freely share it then.
Okay, thank you. Okay, so there are some questions that were submitted about other medications. We're not talking about the FOG medication now. So can you talk a little bit about the difference between like this kind of medication, which I guess we would call a beta-catenin inhibitor, right? FOG would be a beta-catenin inhibitor, and like, or a targeted therapy, and chemo?
Yeah, so I guess technically this is a binder, like it binds beta-catenin to interfere with its interaction with the other protein that's binding DNA. So this is very different. So chemotherapy and most chemotherapies, I should say, bind to either DNA or some other important process within cells to disrupt that DNA and stop the function of that cell. And it turns out some cells can tolerate this better than others. So, and the idea is to have some kind of what we call a therapeutic window where we're killing the desmoid tumor and just killing a few other little cells. And so that's why when people have like hair loss, it's because we're affecting those hair follicles. They're more sensitive than say other cells in the body that don't get damaged. That's sort of more of like a carpet bombing effect, right? We're trying to treat everywhere and we're hitting DNA everywhere. A drug like this is really trying to focus in on that beta-catenin interaction. Now, this drug also binds beta-catenin in normal cells, and that's where side effects can occur. And so what we're looking for here, though, is again a therapeutic window where maybe blocking beta-catenin in the desmoid cells causes them to stop growing, and they're more sensitive than say a normal cell that uses it for another process.
Okay. Okay, so just again, talking about some other therapies. Can you share, or are you aware of any update on the AL-102 trial?
Yeah, so that study completed its phase 3, and I believe it's under data analysis now. I don't know when it's going to be released or what the results are yet, so we're all eager to hear that. But as far as I'm aware, nothing that's been publicly shared yet.
Okay, got it. Did did BBI-608 ever have success with desmoid patients? Did I say that right?
Yeah, you did. Yeah,
BB-160?
so
Okay.
that was an old drug, pre-pandemic. That was in early phase study, very complicated drug. I don't think that we fully understand exactly how it worked, but there were a few desmoid patients who got on that study who did okay. But unfortunately, that drug didn't have the success that it needed to kind of evolve out of that phase 1/2 part. So it's unfortunately not in development anymore.
Okay, great. Now I'm asking about an immunotherapy drug, dostarlimab. Dostarlimab?
Yeah. So yeah, go ahead.
Yeah, I was just going to ask if this type of drug could be an option for desmoid, if you know.
Yeah. Okay. So so immunotherapy, there's lots of different types of immunotherapy, but the general gist is to try to get your own immune system to attack a cancer. And there's different strategies. Some of these are sort of gas pedals that kind of stimulate the immune system forward. Others are more of taking your foot off the brake to let the body's own immune system kind of gear up to attack the cancer. These have been really important drugs in lots of different kinds of cancers: melanoma, lung cancer, head and neck cancer, things like that. In the sarcoma world, including, and I'm putting desmoid in that, it's been sort of less important. Probably, there's certainly some subtypes where they're very active, but in general, probably 10 to 15% of patients will have benefit. What's different about desmoid tumor compared to something like melanoma or lung cancer is that there's very few mutations. So in something like lung cancer that's been started from smoking, there's toxic exposures, many mutations happen in that DNA. For melanoma, it's UV damage from the sun. And all those mutations accumulate over time, and they can lead to kind of new proteins being presented that's not your normal proteins that the immune system can recognize. So they're sort of like immune primed, ready to react to any kind of immune effect. Desmoid's very different, where there's very few mutations, not a lot of new proteins that the body can recognize. They're not typically thought to be inflammatory, where they're kind of primed for the immune system to react to it. So that's a long way of me saying I don't know if there's any data for or anybody who's treated any desmoid patients with immunotherapy, but if I had to predict, I'd probably say it probably would not work. And that's just based because it doesn't have those typical markers that we think about. I'd love to be wrong. So if somebody, in the end ends up trying this, then that would, I'd love to see that.
Okay. There's a question about if any of these drugs are just completely eradicate desmoid tumors, like they just completely disappear. Can you speak to that?
Yeah, so it's hard to know. So in a patient where we give a treatment and it works, we see some MRI findings that are characteristic, where there's certain signals that kind of go from bright to dark, and we think of that as those cells kind of dying, and what's left is more scar tissue. So sometimes if I give a treatment, outside of this trial, just say I'm giving chemo or something like that, and I see an amazing response, I might see that signal go from bright to dark, and then I stop treatment and we kind of just watch it and we see if anything kind of picks up. So that tissue may always look abnormal, and it'd be very hard to know if there's anything alive in there, but we just kind of watch it. That being said, like I've had patients we've done nothing and their tumors grow and then disappear, and we're patting ourselves in the back for not doing anything, which is kind of a funny thing in oncology, but it can happen. So I'd say every patient's unique, and there's definitely some biology there that we're still learning, but probably what's most important is whether the desmoid is growing or causing symptoms. So if it's still looking bright on the MRI but not causing any symptoms and just sitting there, that's fine. If it shrinks to nothing, that's also good. If it stays the same size but it's no longer bright and not causing symptoms, then we think it's probably mostly dead, but no way to know for sure.
Okay. We're going to hop back up. There's another question that came through with regard to clinical trials. So the first question is, how long do clinical trials last? Like if you sign up to be in a trial, how long will that last? And then I have another follow-up question.
Okay, yeah, it's a great question. So this is quite dependent on the clinical trial, but in general, usually in oncology clinical trials, and I'm going to lump desmoid into that, as long as you're deriving benefit, you would stay on the treatment. So say you go on a drug and you're tolerating it well and it's working, you stay on it. You're not tolerating it, having really bad side effects, or it's not working, you would come off. Occasionally, we'll see studies where there's like a predefined endpoint where the study ends after X amount of years. And usually there's some wiggle room there. Like if the study's going to end and you're still deriving benefit, there's a way to stay on it. I've had a couple patients over the years where they did so well, the drug ended up getting FDA approved, and they just basically transitioned from the study to commercial supply, that's a really nice problem to have. And then I've had other situations where like a company's going out of business, and they basically let us have do a specialized trial for the patient for the drug supply that's left, and we kind of do that until it's gone. So there's a lot of different options there, but in general, if you're deriving benefit, you would stay on the drug. Now, the one pause there would be that if there was a drug that maybe has some long-term side effects, there may be a cutoff date of when, you know, it would be recommended to stop. So for these immune therapies, typically the recommendation is to stop after 1 or 2 years, for example.
Okay. And then just one other question. If for the FOG trial, how long, or another study in general, like phase 1, 2, how long are these studies typically open?
Okay, yeah. So also quite variable. So you can imagine during that 3+3 time frame where we're only putting 3 patients on the study, that it will kind of open and close pretty quickly. So we might open to like 3 spots, and there's different ways to kind of dole out who's going to have a spot. But say it goes to me, then I got, you know, say a week or 2 to find somebody to put on the study. And if I got somebody ready to go, I'll sign them on. But if the spots goes to somebody else, then I got to wait till the next 3 till I can find a spot for my patient. So those studies during that dose finding part can kind of come and go. Once we reach what we call that dose expansion phase 2 part, there's typically a lot of spots, and we can, you know, in that scenario, can find a spot pretty easily.
Okay. Let's see. I feel like we're literally at time, even though there's some other questions that came through. That's it. Thank you. Dr. Cote, thank you so much for taking the time to speak with the community. All of our attendees, thank you so much for joining us this evening. We really hope that this was beneficial. I know that I definitely learned something tonight. Just as a reminder, this conversation is not intended as medical advice for any particular problem or as a diagnosis, treatment plan, or recommendation for a particular course of action. It should not be used as a substitute for professional medical advice and services. This webinar was for educational purposes only, and the DTRF does not endorse any treatments or studies. We'd like to thank you for being here with us. After being formatted, this broadcast will live in our video library on our website, dtrf.org. Don't forget to sign up for our newsletter so that you'll be notified when we post it. Thank you so much, Dr. Cote. There are a million thank yous in the chat for you, and we wish everyone a wonderful evening.
Yeah, thanks for having me. It was a lot of fun, and yeah, see everyone soon.
Bye, Dr. Cote. Bye, everybody. Have a great night. Take good care.